So let's look at homework for question 6 now. And this one is a lot of information. And we have the parental generation. You had some purple P and a white P. And this was Mendel's work, right? They crossed them. And it's like, oh, 100% of them are purple. And people were wondering, like, where did the white allele go? And then what he did was, when he selfed this plant, so he took a purple P and selfed it, so crossed it with itself. Then he did the same thing with this, and we ended up with 75% of them purple and 25% of them were white. Okay, so that's the part that I was just telling you here. And then, so let's see. When we looked at this F genes H, it was software predicted genes. And I told you about that F genes H is looking for start codons. So it's looking where are there cues to say this genomic sequence has a gene in it. And then by now you know that the gene is a DNA sequence that is transcribed. Okay, and then how do we know that that sequence may be transcribed? You look at start codons, which would give you cues. Oh, maybe this sequence is translated. And for it to be translated, it has to be transcribed first. Okay, this is stop codons, is end of translation. Then looks for cues for promoters, which are cues saying, oh, the RNA polymerase and transcription factors may bind to this sequence, because the sequence here of DNA has some features that look like it's something the RNA polymerase and transcription factors would bind to. So that's actually most of the work of writing this gene predictor software, is to figure out what sequences are good candidates for promoters. Those parts I told you. The part you didn't know was this part. So it looks at exon-intron junctions, meaning there are DNA sequences that will tell the software that it looks like there is an intron-exon boundary here. So there are DNA sequences that are normally the end of an exon and the beginning of an intron. So that cue is normally a GT in the beginning of the intron and AG at the end of it. Okay, so let's look at this figure. So the A factor, it determines anthocyanin pigmentation in P, and that's what he studied when he was making these crosses. So the A gene encodes what's called a basic helix loop helix. And what is that? That is a transcription factor. So in class, we looked at a gene that encoded an enzyme, the Chalcon synthase, which when we're looking at snapdragons, in this question, we are not looking at the enzyme that may or may not be functional to make the red pigment or the pink pigment. In this case, we're looking at the transcription factor that will then be used on the expressing the gene that makes the pigment or not. So this is whether or not you make this transcription factor will affect the next gene downstream. So it regulates the transcription of the next gene that is involved in the making the pigment or not. So the white flower was most likely a simple G to A transition and a splice site donor. And you're like, what is that? It's this part here that I told you that's new. It's the GTAG. That's the splice site for the intron exon. And then what happens is there's a misspliced RNA, which then makes a premature codon. So this is a copied and pasted from this paper. And this is to show you, like, look, there is a molecular biology side of the genetics that you learned from those crosses. So let's see in figures what that is saying. So there is an A protein, which is a transcription factor that will be responsible for transcribing the gene that will make a pigment. So if the transcription factor is functional, you're going to get the pigment made. So that this protein comes from this mRNA, and this is the full mRNA. Here is where I guess this is where the expression, the transcription. I think this is where the translation stops. So there's a stop codon here. So there will be like more 3' UTR here on the mRNA. Okay, what's important about this spot here is that this is the splice site to tell where to remove an intron or not. So that GTAG that I mentioned up here. So GTAG is that sequence there. So in here. So this means there is in the DNA sequence a GT here and an AG here. And for the functional transcription factor, that part gets cut out. This 5,775 base pairs gets cut out. But for the little a allele, so in the DNA sequence, instead of having GT, you have an AT. So there is a one base change, which then makes this intron be different. So you don't remove this part, and then within this intron, there's another GT, and that's when the cell recognizes, oh, I should cut here, not here. So then what happens is that there is this extra piece that originally was intron, but now it's an exon, but then there's an early termination here. So instead of terminating over here, you terminate it here. So your transcription factor now is shorter. So in conclusion, this is the transcription factor that would act on the transcription of some enzyme that makes pigment. And then a change in the splice site leads to alternative splicing. So you have a different mRNA molecule that the translation gets terminated early. And then this transcription factor here, because terminated early, then it doesn't bind whatever is the gene it needs to control the transcription of, then you have no pigment made. That's the story about the purple and the white pigments, which is different from the white, pink, and the red, or that I talked about in Snapdragons, because that one was incomplete dominance, and that had to do with the enzyme activity. In this one, it has to do with not the enzyme activity that's making the pigment, but the transcription factor that is controlling the transcription of some other gene. Okay, so in the white flower, which has the genotype little a, it changes the splice site. So this is everything I was just telling you about here. Okay, and this is just curiosity because I spent so much time reading this and to tell you this story that I had to write to tell you about this. All right, let's go to the questions. And then I'm going to ask you to help me here whether or not it's true or false, so that I see that you are participating, because that was kind of a long story.